Welcome to the Personalized Medicine podcast. This is the place where scientists, clinicians, and entrepreneurs discuss the progress of this rapidly developing field.
I'm your host, Luisa Hallmeyer-Wacker. Let's get started with this week's episode. Significant progress has been made in several fields of medicine towards personalization. We've addressed some of these in previous episodes. Today, I want to examine how personalization has been incorporated in reproductive medicine. It is my great pleasure to welcome Dr. Helen O'Neill, an expert in the field of reproductive science, on the podcast today. She's a lecturer in reproductive molecular genetics and the director for the Master's Program in Reproductive Science and Women's Health at University College London. Her research focuses on pre-implantation embryo development and the use of genome editing to assess understanding and treatment of disorders of infertility. Beyond her work as an academic, she recently co-founded Hertility Health, with the aim to provide women with an easier way to get personalized insights into their reproductive health. Helen, thank you very much for finding the time to talk to us today. I look forward to our conversation and to learning more about your work as a scientist, educator, and entrepreneur.
Thank you. I look forward to speaking to you too.
So you hold an honors degree in genetics, a master's in prenatal genetics and fetal medicine, and you obtained your PhD in the Department of Stem Cell Biology and Developmental Genetics. It seems like you've had an interest in genetics and more specifically in reproductive genetics for a long time. So what got you interested in this field of study in the first place?
I think my interest stemmed from the moment I was born. I am an identical twin. So the idea about genetics and how I am similar, in fact, identical to my twin sister has always been a driving fascination for me. I always looked at her and thought we looked so different. I always thought we behaved so differently also, and yet everybody else looks and sees the exact same thing. So for me, the idea of studying something that would give me insights into what makes us so unique or makes us the same has always been in the back of my mind.
Yeah, that's for sure a very, very unique story on why you study something or why you're interested in something. So let's dive right into reproductive medicine. When a lot of people hear about reproductive medicine, they think IVF, infertility, and women's health. So as a researcher in this field, how would you respond to that?
I think that there's a lot of truth in that, but the field of reproductive medicine is so huge. It has really propelled our understanding into women's health, but more specifically obstetrics and pregnancy and the outcomes in which we can now look into the mother's womb during pregnancy and see the fetus as it grows through scanning, but also test that fetus through means such as putting a needle in and taking a little piece of tissue. But equally now that has evolved into non-invasive prenatal testing. So I feel, yes, there is a lot to do with reproductive medicine and its association with in vitro fertilization and fertility, but also there is a huge field that must not be forgotten in obstetrics and newborn screening and in utero screening also. And maybe the next generation will be editing or looking at testing the germline.
Yeah, and well, that's something that you've focused your research on as well, looking at genome editing, potentially germline editing, and CRISPR technology has really revolutionized that field. So could you go into a bit of detail how the ability to precisely edit genetic material has changed reproductive health? And maybe a second part to that question is, will this technology lead to a more personalized approach in the future?
Sure. I feel that genome editing as a technology is one that is beautifully positioned to be embedded into any form of research, any form of science, and any form of medicine. It has propelled our ability to interrogate the genome at specific and precise locations. And it has also improved our understanding of diseases that previously we had very crude methods of understanding. I guess the most basic way of saying that we can use genome editing is to compare it to previous methods. So previously we would almost have to wait for a disease to pop up in the genome, and then you study how somebody is affected by it. So when you study how somebody is affected by a disease, you can look at their own genome and say, where in their genome has there been a mutation, and can we ascribe this specific mutation to this specific disease. So for that, you need multiple people with the same disorder. And once we've found out the specific gene that could be involved in causation of a disorder, then we can start to interrogate that gene. Now, the ways in which we can do that are very time consuming. I spent many a late night in the lab trying to create mutations in cell lines. The work that, for example, took me 2 years to do during my PhD would now take approximately 3 to 4 weeks, which is a little bit disheartening for me after spending so long doing genome editing the difficult way, but it's also very exciting for the next generation that we can now propel our abilities in the lab so much further.
That gives us a very good understanding of the scale that CRISPR allows us to look at in terms of genetics and in terms of causation for disease and potentially treatments. How would that translate to reproductive health specifically?
So there are a number of ways in which this can translate to reproductive health and reproductive medicine specifically. I guess one of the more controversial areas is an area known as germline genome editing. In other words, that you would use CRISPR genome editing to actually edit a human in its earliest form. I guess you can't call it a human in the earliest form, but edit an embryo. So you can use this it could be used as an adaptation to current testing methods. So when you mentioned that I work in pre-implantation embryo development, what that essentially means is that when a human egg is fertilized by the sperm and it starts to grow, prior to the point where it would implant, it is known as pre-implantation. I'm making the word pre-implantation. I'm spelling out the word pre-implantation for you here. But I guess it's essential to visualize that, because prior to the implantation of the embryo into the womb, we can actually grow that embryo outside of the mother's body. So it means that we have amazing ways in which we can study and learn about the earliest form of human development. Now, if you think about the ability to grow a human embryo prior to implantation, well, there are a number of ways that we can study that embryo and potentially edit it. So when I speak about the more controversial forms of CRISPR genome editing in the germline, this is what I mean, that you would be able to take this early embryo prior to its implantation, and if you suspected that it had a mutation or was carrying a certain disease, that you could use genome editing to specifically go into that embryo and correct that mutation prior to it implanting and prior to it growing inside the womb. Now, how this is perceived is certainly it's a controversial area. Some people believe that we should not be editing any human embryos. Some people that believe that where you could also use this is to edit the sperm or edit the egg. Those are the progenitor cells. And yet there are others who feel that there are plenty of things that we are doing already in the field of reproductive medicine, in the field of IVF, that are technically and indirectly editing those embryos anyway. Essentially, I feel it's so important that we do not neglect the epigenetic changes that are happening in a very dynamic genome. So the early embryonic genome is so dynamic. It's constantly changing, altering. It goes from using just the maternal or the mother's genetics to help build its early steps to using its own genome in a process called embryonic genome activation. And when you put an embryo, which would be developing inside the uterus, and you take it out of the uterus and you grow it in culture media conditions in a dish, and you grow it in an incubator, all of these exogenous forces that are not innate are bound to have transgenerational changes or just generational or genetic changes on the embryo itself. It is we can try as much as we want to mirror the uterine environment, but this, as you can imagine, is a very difficult thing to do. So in terms of editing a situation or editing the epigenetics, I think we're already doing that. But again, people find a lot more of a problem when it comes to making purposeful changes in the genome.
Yeah, I think that for a lot of people, it brings up kind of fear of a dystopian future, where we start with editing potential diseases and end up editing more and more of things that are potentially not diseases or even enhancement. So I would like to dive a bit more into that from your perspective. Yeah, what are the ethical concerns that you see in the scientific community and in society, and have they in some way changed the way you conduct research?
Most certainly they have changed the way in which I conduct research, because you cannot separate the ethics and public perception of things. There is a trickle-down effect when it comes to public perception of a scientific practice, and we are ruled and bound by public perception in terms of our ability to pursue certain areas of reproductive medicine and also receive grant funding for that. So I think, unfortunately, there's far too much of an emphasis on dystopian futures and unwelcome forms of the use of genome editing. This is the type of argument that comes up time and time again. It came up when we first studied embryonic stem cells. It came up when we first had the ability to clone. It came up back in the 70s when we started using recombinant DNA technology. The same arguments were used, whereby people were able to project their fears and project their ideas about what it could potentially be used for, and focus those fears and focus those potentiality into negative aspects of a technology. And the truth is, we are bound by ethical norms, we are bound by ethical laws, and we are bound by real laws, and we are unable to pursue those routes, nor do we wish to. So unfortunately, I feel like the ability to convey information about what we can and cannot do is so important. So it's really important for me as a scientist and a communicator to communicate my science, communicate my desires as a scientist, and especially in this field, to be fully transparent about my desire to do good. And I feel that every single scientist I know has the same ethos, that they really feel that there's so much potential for genome editing, not just for discovery into diseases, but discovery full stop. We know less about the early human embryo than we do about a mouse embryo, than we do about a Drosophila or fruit fly embryo, or a fish or a worm. We have been able to dedicate more research into those because of the ethics that is involved in studying early human embryos and I suppose embryos in general. And yet there is so much for us to learn, and genome editing really could open up the field for us and to help us to understand the current black holes in our knowledge.
Yeah, and I think now that we've addressed the ethical concerns and maybe started with the negative side or the more critical side first, I want to give you the space to talk about some of the applications of CRISPR that you find especially exciting. So I know that you've worked on CRISPR genome editing for the treatment of sex chromosome and neuromuscular related disorders. So could you talk about an application of CRISPR that you've worked on and that you see evolving over time and that you find especially exciting?
Sure. So my research has been actually predominantly focused on the safest ways that we can deliver genome editing components into an embryo. And when I speak about my research, I think it's important that my obsession is safe practice. So as I said, there are many things that we have yet to learn about early human embryonic development. But there's also very many things that we have to learn about genome editing. And genome editing is seen as a technology, but I think it's very important to remember that it is a collection of technologies, and we are using this collection of technologies on a very dynamic, as I said, form of biology, and that is human reproduction. So we're essentially working with two very quickly moving targets. The first is quickly moving in the field of genome editing. That is moving in terms of its progression, and the other is moving in terms of its dynamic growth as a one cell that turns into 200 plus cells. And so I think we have to take things one piece at a time. The first thing that I have focused on is the timing of delivery. Another aspect that I focus on is the method of delivery. So whether we use needles to inject the nucleic acid into the embryo, or whether we can do electroporation and shock the cell membrane to successfully put in the genome editing components into an embryo. The other methods that I have looked at are the effect of mosaicism. So mosaicism is a word that is constantly used in the field of assisted reproduction, and it's usually associated with aneuploidy. So when you have mosaicism in an embryo, you're usually talking about how many of those cells in that embryo have a normal copy of chromosomes versus those that have an abnormal quotient of chromosomes. And generally, if you test a clump of cells in an embryo and some of them are found to have a normal amount of embryos and the other cells are found to have an abnormal, that embryo is then known as mosaic. But the type of mosaicism that I am interested in is if I am to make an edit in an embryo at the 1 cell stage, and that embryo continues to grow into 2 and 4 and 8 cells, and the edit that I have made only stays in one of those cells, or it only goes to 2 or 3 of those cells, then that for me is CRISPR-derived mosaicism. So I think there's an important distinction in those two, but I'm very interested in how we can replicate or successfully ensure that the edit in 1 cell goes into every single cell, so that if you were to correct an embryo that had a mutation for a very debilitating disorder, that we could ensure that that embryo was free from the disorder for transfer, for implantation.
Yeah, and as I understood, if you have a CRISPR-derived mosaicism, then that wouldn't be safe, right? Because you would create an embryo where part of the cells, or a fraction of the cells, would have the alterations and the other wouldn't. How do you, from a laboratory perspective, how do you test that? How do you work on these technologies?
It's again through a collection of technologies, but none of them are perfect. When it comes to, I feel the CRISPR side, or the genome editing side of things, is moving faster than ever, and yet the embryological side of things is a little bit more slow moving. And I say that because it is due to the physical difficulty of doing research on human embryos. And that is largely to do with ethics. And then there's the physical difficulty of disaggregating a human embryo into individual single cells and testing those single cells. I think the third important factor to acknowledge there is the cost involved in sequencing, which, albeit that it is plummeting, it's still very expensive as a researcher to do that.
So you do sequence the entire genome or the entire single cells to make sure that there's no other alterations that have happened? Or could you go into a bit more detail how you use sequencing in general?
So what we do is we take a clump of cells from the embryo and hope that they are representative, which is again, there are multiple ways of hoping that we can get proper and reliable insights into an embryo. One of the evolving fields is a non-invasive way of looking at embryo development, and that is by looking at the culture media. If you can interrogate the culture media that the embryo has been growing in, we're gleaning more insights into testing an embryo non-invasively. So in other words, that we wouldn't have to rely on taking a big biopsy or a clump of cells from the embryo. at the blastocyst stage.
So it's been interesting to hear about your research and what you've been focusing on. In your opinion, out of this, or in general, in terms of genome editing and gene editing, what would be the first technology that would be translated into clinical practice?
Well, there are those who feel that current methods for testing embryos are limited in that while we can reliably test embryos using pre-implantation genetic testing methods, and this involves taking a clump of cells, as I mentioned, and analyzing them, there are others that feel that this is insufficient, not necessarily due to the testing process, but due to the lack of sufficient embryos. Let's say you have 10 embryos and 5 of them are carrying the mutation, so you can't use those, and 4 of them do not develop sufficiently, and you only have 1 left for transfer, which fails. Then you're left with no embryos left and no future child. If, on the other hand, you took those 10 embryos, the 5 that contain the mutation, you could correct that mutation and you have essentially double the number of embryos that you have available for transfer. This is definitely a technology which is controversial for sure, but it is also, I think, a real eye-opener, and it is giving hope to a lot of families who are carrying rare and devastating diseases.
Yeah, it's been really interesting to hear your insights into reproductive health, genome editing, or germline editing more specifically. And I know it's a very controversial topic where a lot of people have ethical concerns or other kinds of concerns. But I think it's an important topic to talk about nonetheless, because the technology is progressing or even there already. And it's important to educate both the scientific community as well as individuals about what's possible and both risks as well as benefits.
We are doing this show for you, and your feedback is very important for us. So if you have any suggestions or comments, would like us to cover a specific topic or recommend a guest, please write us an email to team@pmedcast.com. Or you can reach out to us on LinkedIn, Twitter, or Facebook. Just type in Personalized Medicine Podcast and you will find us there. To download the show notes for this episode, visit our website, pmedcast.com. It's pmedcast.com. The show notes include guest bios, links to their most notable work, and recommendations for additional reads on the topic of the episode. Make sure to check them out. And don't miss the next episode. Subscribe to our podcast on your favorite podcasting platform. Give us a rating and leave a comment. It will help us make this show better. And now, let's get back to the interview.
Next, I want to focus a bit on your work as an entrepreneur. So in early 2019, you co-founded Hertility Health, which aims to provide women with an easier way to get personalized insights into their digital biological clock and reproductive health. So what led you to launch the startup?
For me, it was as much a personal endeavor as it was a professional endeavor. Working in the field of reproductive medicine, would almost weekly have people, friends or friends of friends, contact me about their own reproductive health. Women are very fortunate in that we have the incredible capacity to carry life, but we are also unfortunate in that we almost 50% of women suffer from some form of gynecological pathology, and very often that gets ignored or it gets mistreated. And for the most part, women who have problems when it comes to their periods or abnormal cycles or pain are just put on the pill. So they end up masking their symptoms. And we are definitely a group of women, or a generation of women, who are putting our careers first. And so we deprioritize our reproductive capacity and our reproductive needs to the point where more women than ever, in fact, I think double the number of women, are childless over 40 without choice. Now, if you choose not to have children, then I salute you. But if, on the other hand, you have always wanted to have children and you are not in the position yet to do that, then it's a very difficult time, because we are not prioritizing women when it comes to having a family. We are now expected to be the breadwinner, the bread baker. We are expected to do many different things now. And unfortunately, having time to have a family is not one of those things. So we essentially wanted to set up Hertility Health to enable women to check in with their fertility and check in with their reproductive health. We do this by allowing you to check from the comfort of your own home by sending just a reproductive health kit. So it's just a simple kit that you answer a questionnaire that we have clinically curated. This questionnaire, we've worked with endocrinologists, specialists in fertility and reproductive medicine, and ourselves to create this questionnaire, which will allow us to understand which hormones we should test you for.
And if I understood correctly, you founded this company also with your twin sister. Is that correct? I just thought that was interesting, because that kind of goes back to the very beginning of our interview.
It's funny, we've come along on it. We've definitely started on very different journeys. I actually founded this with a colleague of mine who is an ovarian biologist, Dr. Natalie Gatroy, and she and I both had the same frustrations about not being able to check in on our own reproductive health, despite knowing the risks ourselves, and we were putting the risks, which is the weight game, forward the whole time. And we began this, and my sister is a lawyer who works in the corporate world. So we were constantly vying for her attention to help us out. And when she came on board, she really felt like the mission was something that was so worthy of her attention that she has joined the team. And so I feel very, very fortunate to be, after many years of doing very different things, working alongside her now.
Well, it's always good to get, I think, input from a lot of different areas. And I think having someone from the corporate world helps a lot of scientists make the jump into entrepreneurship. Just to circle back to Hertility Health and the service that you offer. So you have this questionnaire. Do you collect any kind of biological sample as well?
Yes, of course. So essentially the questionnaire is just to help us understand your health needs. So we call it a care questionnaire, which we have curated to enable us to glean insights into your reproductive history, your cycle history, and your own biometrics, essentially. And from your answers, we will then know which hormones to test you for. So it's very much a tailored test. We test all sorts of hormones related to your reproduction, but also your thyroid function, as these are so interconnected. And once we've understood your answers and we know which hormones to test you from, then we will send you a kit to your house, and you will do a simple blood test. So it's just a finger prick, and we collect some blood, and you put it in a prepaid mailer, and it goes to our accredited labs, and then we are able to give you insights into your reproductive health. The important thing is that we have really struggled to understand how it is that reproductive medicine has come so far, and yet when it comes to giving people answers about their bloods and results, the way in which we deliver this to patients and to people is so limited. It's the majority of people don't understand what it is they're looking at. The results are displayed in picomoles per milliliter, micromoles per liter, and we've essentially worked very hard to make this process as understandable as possible, so that everyone can see what is the hormone, why do we test it, why are we testing you for this, and what does your result mean. So everything is explained in a very understandable format on a dashboard, so that you're able to track your symptoms, track your cycles, and from there we'll give you advice on anything that you would need, whether it's egg freezing, or whether you're contemplating having a baby, or whether it is that you need support from an endocrinologist, or just to be happy to know where you're at.
And one clarifying question for that: if you're looking at hormones that change throughout the cycle, how do you deal with that if you have a single blood sample at a single point of time?
So we ask you to test on day 3 of your cycle. So you're asked to test on a specific day, unless we're testing progesterone, in which case we'll do that at day 21. So there are explicit guidelines on what day to test.
Okay. No, I mean, I did part of my PhD. I also collected blood, and I tried to test hormones, and it was from animals, so it's a bit different. But we had that issue that it was always difficult to determine what part of the cycle they were in using only the hormones. So I was more from my personal interest.
Okay, great. Well, truthfully, that kind of leads to why it is that we, previously women have not been prioritized when it comes to testing at all because of this. Even animals, we use male animals when it comes to research because they don't have the estrous cycle. So this is, again, I guess why research into women's health is in its infancy, is because women have been excluded in clinical trials and testing for drug development and testing in general. So we are creating the world's largest database of female gynecological pathologies. We are really hoping to replace the why with, well, because when it comes to all of the different symptoms, all of the different, individual experiences of women through their cycle.
So I hate to play the devil's advocate.
Please do.
But so as far as I understand, the current focus of Hertility Health is on female health and fertility. But there's always two sides to the coin. And so isn't it also important to include men in fertility education and potentially even testing?
Absolutely, yes. And in fact, we've come so far in our understanding. Previously, people always ascribed infertility issues to being a female issue, and it's so far from the truth. It went from statistics showing that 80% of infertility was female related to 60% of infertility is female related. And now we acknowledge that 50% of infertility is male factor infertility. And so I couldn't agree more that we need to include men in the discussion around fertility. We are certainly dealing with that with Hertility Health, whereby we are also offering a male hormone testing panel. They won't get the same pink box, but yes, watch this space.
So it must be quite hard launching a startup with this current COVID situation, especially because you're based in the UK, which is currently still on lockdown. So how has this influenced the startup in general?
I'd like to say that COVID-19, albeit that it is a very difficult time, has actually offered us some silver linings. We are offering the kits not just to any individual woman who wants to be tested, but also IVF clinics. So we have partnered with IVF clinics so that for the women who cannot attend their appointments, quite naturally, in person, they can still be tested remotely. So this has actually worked well for us to try and rally behind and support some of the IVF clinics that are not able to see their patients. So I feel that it has been quite helpful to women who are desperate to be tested and desperate to start their treatment in assisted reproduction, that we can actually come in and help them.
It sounds promising, and also like a good opportunity to make contacts within clinics and convince doctors that this is a useful thing.
Yes, they have time. Yes, they have time on their hands now to listen to us.
Yeah, which is always difficult with clinicians.
Yes.
So what are the next steps for this startup?
We are hoping to grow. One of the main drives of this startup is research. So we are founded by researchers, and we are absolutely dedicated to really improving the lives of women through further education, through further research. When I said we're building the largest data set, what I mean is that these insights that take your biometrics, that take your personal history, your cycle history, your medical history. These are really critical in ascertaining the overall outcome when it comes to your ability to conceive. So often we only take a unilateral view of somebody's health by just looking at their hormone results or just looking at their medical history. And to me, it's fundamental that we collect all of this, and using all of it together, we can build a more promising picture for how we can determine, I guess, your fertility potential is only one small aspect of it, but also your reproductive health in general. We are running a clinical trial at the moment, which is looking at 4 different gynecological pathology units. One of them is for polycystic ovarian syndrome, one is endometriosis, the other is pelvic pain, and one is recurrent miscarriage. And we're really hoping to improve the current diagnosis time for conditions like endometriosis, which is shockingly 7.5 years. And we're hoping to bring that down to 7 days.
Yeah, that sounds exciting. And also, I mean, as a woman myself, I think something that's really necessary to have a more open communication, and then as well as testing, in order to improve female health or women's health in general, not just in terms of pregnancy and infertility, but also in terms of information, especially for young women.
Right, exactly. We're really hoping to reach a much younger audience with this, that, you know, all women should care about their reproductive health because it impacts so much of your day-to-day life.
It's been really inspiring to see you or hear about your success, both as an academic, a science communicator, and an entrepreneur. So what has it been like to transition between these roles, and what have you found the most challenging, especially in this new chapter of being an entrepreneur?
For me, it's been really exciting to be able to tie in the things that I'm passionate about, and that is research, that is communication about my research and communication about science in general. I feel very lucky that I'm able to do all of the things that excite me and motivate me and bring them to the public through lecturing, through public engagement, but also with Hertility and being able to bring reproductive health education and information into the hands of every woman, and not just be heard by my scientific colleagues or not just be heard at research conferences. So for me, it's been an enjoyable transition because I've just embraced the ability to tie all of my passions into one big knot.
And potentially reach a larger audience.
Yes, exactly. That's exactly it.
It's been really exciting to hear about Hertility Health. To end today's interview, maybe one last question, which is a big question. So what's your outlook on the future of personalized medicine and reproductive health? So where will we be in 10 years?
That's a great question. I feel very passionately about personalized medicine. I think for far too long we have generalized when it comes to treatment and when it comes to studying disorders. For me, the use of a number of technologies in one are going to propel personalized medicine. So that is genetic sequencing and the use of genetics in medicine, and genome editing, but equally our ability to use machine learning to interrogate that data. I feel like it is so many fields that are propelling personalized medicine forward in a way that previously we just couldn't have done before. So it's almost like a tripod effect, and through those 3 methods, with genome editing, with sequencing and interrogation of genomes, and also artificial intelligence and the ability to look at data in previously unforeseen ways, this is what gives us a very strong standing point from which we can build upon. I'm excited about the next 10 years.
Yeah, so am I. Well, thank you, Helen, for sharing your insights and expertise on reproductive health, on CRISPR technology, on genome editing in general, and your startup on Hertility Health, and a little bit on women's health advocacy as well.
Thank you.
Before we end today's episode, could you let our audience know how they can reach out to you and find out more about Hertility Health?
Sure. You can reach me at helen.o'neill@ucl.ac.uk, or you can find Hertility at hertilityhealth.com, which is fertility with a H.
Thank you for listening to this episode of the Personalized Medicine Podcast. If you like the show, please subscribe, rate, and review our podcast online, and make sure to share it with a friend. And don't miss out on the next episode yourself. Subscribe to the Personalized Medicine Podcast on your favorite podcasting app. You can find us on Apple Podcasts, Spotify, Pocket Cast, Stitcher, and many more. To access the show notes for this episode, visit our website, pmedcast.com. Our show notes include guest bios, links to their most notable work, and recommendations for additional reads on the topics of the episode. Engage with us on social media, where we share news and exciting content on personalized medicine. You can find us on Twitter, LinkedIn, and Facebook by typing in Personalized Medicine Podcast or using our handle, pmedcast. And if you have any feedback or would like to suggest a guest for the show, write us an email to team@pmedcast.com. Have a great day, and until next time.